Okay, guys, let's get started. It's not, we're going to hold 10 people today. That's fine. We'll just still record it and post it for everybody so that they have it. And these chapters go by a lot faster on these reviews than the other ones too, because I've updated them more recently. These reviews have like a lot less complicated questions on them, so it should be less time. So we'll be going over chapters 19 and 20 today. We'll give like a little mini review of what was on our study sheets for those guys, as well as our missed topics as per usual. So we'll start with chapter 19. Remember, this is our skin and eye diseases. The skin and eye have very similar biota associated with them, but the eye is going to have that immune privilege, which we're going to get into more. We got into it more in our talk for chapter 20, but also in our review for chapter 20, we'll talk about immune privilege again. But both of these guys have their own defenses. So the skin is going to have its high salt. It's going to have the low pH associated with it. And that a lot of that is due to the arid environment, the fact that we're exposed to air in general. So it makes it dry, it makes it arid. And the organisms that can grow in those environments will create byproducts that lead to that lowered pH. They also have to be able to put up with the high salt content. So they're like halo tolerant is what we call that. Lysozyme, our cells will produce lysozyme in our sweat, in our tears, and our saliva to help break down peptidoglycan walls of our bacteria that could be invading. We also have keratin, our waterproofing protein, that helps create that solidified barrier at the surface of the epidermis. And then the normal resident biota of the skin that we are most concerned about. The top two are going to be Staphylococcus epidermidis and Propionibacterium acnes. All right, so moving into the diseases, we discussed some of these major ones. I'm not going through every single detail of every one again, of course, but we talked about strep pyogenes, basically being like the initial instigator of impetigo, and that later on Staphylococcus aureus will come in and have its exfoliative toxins like lead to the development of that flaking aspect of what we see with impetigo on the skin. Then we have staph also being associated with MRSA, of course, because that stands for methicillin resistant staph aureus. If it's resistant to methicillin, it's resistant to antibiotics that would be used leading up to the use of methicillin. So base level penicillins and things like that. Next, we have gas gangrene. Of course, this is going to be a deeper tissue infection. We've skimmed right over cellulitis, but that'll be a tissue infection as well. But gas gangrene being caused by Clostridium species is going to be a gram positive. Remember, because all of our, I can't with the dogs right now, all of our endospore forming bacteria also happen to be gram positive. So there's two, like I said, I told you guys, I warned you I'd be saying this over and over again in the beginning of the semester, but our our endospore forming bacteria, all of them are gram positive. And the only two that we need to know about are our bacillus and our Clostridium species. So those guys we know have to be gram positive because endospore forming bacteria are gram positive. So Clostridium perfringens has to be gram positive because it is endospore forming. It gets into the deeper tissues. It leads to the development of gas gangrene. The bacteria themselves being anaerobes, they do produce gas as a byproduct of their normal anaerobic respiration. So that'll build up in the tissues, cause pressure in the tissues, and lead to release of fluid and gas from the tissues themselves under the skin, creating blisters, and those blisters can rupture and the skin can rip as well. But anyways, that lead to myonecrosis and other issues. So that's Clostridium perfringens specifically. And it'll pop back up again when we talk about GI food poisoning, because it makes a toxin associated with like consuming certain types of foods as well. It's a bacteria that's found pretty much everywhere, like a lot of the Clostridiums are. All right, next we have chickenpox. It's caused by a human herpes virus 3, also known as varicella zoster. So it's going to hide out in your nerves and can get reactivated later in life as shingles along the nerve that it hit on. With smallpox, we have a virus that causes smallpox. This is called variola. There's a major and a minor version. One that we think most commonly of is the major. It's pretty high mortality rate. You'll have a normal like feeling ill as well as a very disfiguring rash. It's very obvious as well. And we have a vaccine to thank for that. It was the very first vaccine ever made. It was made from cowpox, which we call vaccinia, and why vaccines are called vaccines. So cowpox itself is caused by the virus vaccinia. Smallpox is caused by the virus variola, but because they are so closely related, we were able to create a cross-reactive immune reaction. And that's been a super lucky thing because that was a horrible disease. And we're all very glad that that's been eradicated. Next is measles. Measles is associated most commonly with a rash. It's also a viral infection, but it can progress into meningitis, or encephalitis. And those are things that we have talked about previously, or I say previously, but in chapter 20. Ringworm is a fungal infection, despite being called ringworm. There are no actual worms involved. It's just a round lesion that's typically associated with the version that appears out on the body. The corporus, the tinea corporis. A lot of these diseases are called tinea, which means worm in Latin, but they aren't associated with worms. They're all fungal infections, making them mycoses. And these dermally related or cutaneous mycoses are caused by dermatophytes, which are fungal species that are associated with skin infections. So there's a lot of different species, at least 39 different species that can cause ringworm, and it includes things like typical ringworm and athlete's foot. Conjunctivitis, we have inflammation of the conjunctiva, so our protective layers of the eye. This can be bacterial in origin. If it is, it'll have a milky or yellowish discharge. Viral and allergic, we have clear fluid that'll be exuded from the eye tissues, and then all of these are going to have redness associated with them. We typically call this pink eye. Trachoma is an eye infection that is going to lead to blindness. It's the leading infectious cause of blindness in the world. It's caused by the bacterium Chlamydia trachomatis. And then river blindness is another blindness that's infectious, but this one is caused by transmission of worms into the eye via flies that are going to transmit those larvae into the eye tissues, and then they migrate into the eye cavity and stay there. The pathogenesis of this is mediated entirely by the fact that the worms themselves are infected with a bacteria called Wolbachia, making it an endosymbiont. So it lives inside endosymbiont because they have their life cycles kind of reliant on one another. This is true actually for a lot of our helminth infections. All right, chapter 20 was our nervous system diseases. I didn't change it. This is supposed to be 20, but you guys don't care, I'm sure. Anyways, so our normal biota for the nervous system, we shouldn't have one. It should be sterile. There should be no bacteria or viruses or anything that are normally associated with infection of these tissues. So our normal biota should be non-existent here. Our nervous system exhibits immune privilege as well. We don't want overactivation of our inflammatory response because the inflammatory response is damaging to tissues. So we don't want to damage your nervous system because you can't get your nervous function back in your central nervous system if there is damage done. And it's actually harder to heal any nerve damage, even the periphery, than it is in any other tissues in the body. So we want to be sure that, and still maintain function anyways. So we still want to be sure that we are avoiding inflammation wherever possible. So we have our own immune cells that will stay stationed there, our microglial cells, and our brain macrophages. These guys have a reduced inflammatory response. And if we have any crossover of our immune system into the central nervous system, like our white blood cells, like our lymphocytes and stuff, that's only going to be in severe situations. So we're going to want to keep track of that sort of stuff there. Sorry. I'll love you guys.
Yeah.
And I slid it right.
All right. Anyways, what did I just do? No, I can't get back to where I was trying to go. We'll just go ahead and do this to everybody just to be safe. Okay. All right. Anyways, so immune privilege, we talked about that. So moving on to meningitis, we know this is a syndrome. It's a syndrome, or what we call an anatomical syndrome, because it's being defined by the symptoms and signs that are associated with the disease and not necessarily by the cause. So with meningitis, our signs and symptoms would include things like photophobia or light sensitivity. It would include fever, malaise, pain and stiffness of the neck, headache, and things like this. And if you have these symptoms, we would call this meningitis. If you have inflammation of the meninges, it's meningitis. So there's a great variety of causes of this. And we went over this actually pretty well. I made a little chart for it in my night class, but it essentially is the same exact little outline that we have here that I had written on that board. So we have our bacterial meningitis. The first one that we went over in class was actually Neisseria meningititis, which is the worst one. So I actually want to, I don't know, remember how to do this. Let's see, here we go. Not that one, this one. Okay, so we have for bacterial, Neisseria meningititis. This one is gram negative. It is a diplococcus. So here, if we look at this in a microscope, we're going to be looking at some red butts. I know that's crazy, it's what it looks like though. So we have round cells that are in pairs. So diplo means pairs. And coccus, we know it means round. And those together, paired little round cells look like little tiny butts. So we have our little red butts in our microscope there. We also call this the meningococcus. Cool. So, so that's one of our bacterial meningitis. This is also the most deadly of all of them. And our meningococcus, when it causes meningitis, it leads to the development of a rash as part of its presentation. This is a petechial rash. These are little tiny burst capillaries. That leave little tiny spots under the skin that look like a rash, but it's actually burst blood vessels. When this spreads into the bloodstream, it leads to meningococcemia, and that is an incredibly fatal disease. So we want to be sure that we're getting this, as bad as the meningitis is on its own, we want to be sure that we're getting it under control early on. All right, so that was our first one we talked about in class. The second one we talked about in class was Streptococcus pneumoniae. And this guy is a gram positive. And this is also we know it's a coccus, it's round, it has it in the name. We know it's strepto, so it's chains. So chains of round cells. So I'll put chains. So gram positive chains of round cells. So on this one, we're going to have purple chains. In our gram stain. All right, so this one's the most common communally acquired one. So this is anything that you're going to get just from being around people, as opposed to like the healthcare associated diseases that we'll talk about later on in the unit. So this one is most common. And it's also known as the pneumococcus. So number 1 has a vaccine. Number 2 has a vaccine. And number 3 will also. This is definitely a 3. So this is Haemophilus influenzae. And for what it's worth, yes, the way that I actually am writing these out on here is the correct spelling for them, if you guys are concerned about that. So Haemophilus influenzae, and then we're going to have Listeria monocytogenes after that. So Haemophilus influenzae, this guy's going to be gram negative coccobacillus. And these guys are going to be red pills, essentially. Red, squatty little pills in their gram stain. We do have a vaccine for this one as well. We also call this the Hib. I don't know why this is wanting to not let me write. I guess I can't actually write on that part of the slide. That's nice. I wish I would have known that before I started writing. Okay, whatever. So we'll just continue it over here, I guess. Let me, so that this is clear, because I don't want this to get confusing, because now I'm going to have to make a different slide for the other stuff. What I want is an eraser. Here we go. Okay, so let's rewrite that on a different area. It's fine. Okay, so number 3. All right, so we said it is a gram negative coccobacillus. Coccobacilli are like oval shaped, so they're kind of between round and rod. So that's why I say they're like a pill. So, or we could say oval. It's kind of just whatever you prefer. But so this guy is going to be a red, let's say red oval. That's fine, whatever. Or pill, whatever. And it's also known as Hib. And that whenever we are getting older, this is, or if you're little babies, or if you're immune compromised, then they'll push the Hib vaccine. But there is a vaccine for this one as well. All right, and then our 4th one for the bacteria is Listeria monocytogenes. All right, and this is all meningitis. And this is just the bacterial meningitis. So Listeria monocytogenes, we usually get this from food. It's going to be dairy and meats and certain things that are like on that spectrum of the food. Sorry, I'm getting all these emails. Great, thanks, watch. Okay, anyway, so it is a foodborne illness, and this one is a gram positive, and it is also a coccobacillus. So automatically with this one as well, in our gram stain, we have purple ovals. So we've got our 4 major causes of meningitis easily identified completely and entirely by gram stain. That's usually our main form of diagnosis. We will get a lumbar puncture, and then our gram stain, the CSF directly. We don't even culture it first. So remember, CSF is your cerebrospinal fluid. It provides nutrients and cushioning to the nervous system. So this is foodborne, and there is a 1 in 5 chance of fatality of the unborn fetus. So 1 in chance fatality of the fetus if the mother is infected. This likelihood of fatality increases if the mother is exhibiting any symptoms. And fatality is about 70%, I believe, if there is actual presentation of the disease symptoms in the fetus, or in the baby after it's born, something like that, but it's pretty high. So this is a very, very serious thing for pregnant women. So we don't want to take any chances here. So 1 in 5 is the same thing as 20%. So what else am I missing about this? I don't think really much. It is modal for, and it's an intracellular pathogen, so it gets inside of the cells. Which is how it gets across the blood-brain barrier. Blood-brain barrier literally just meaning that we have a tighter stringency for what crosses out of the bloodstream and into the brain based on receptors on cells, based on tighter junctions between the cells, as well as thicker cell layers on the actual vessel endothelium as well. So all of that manages what goes in and out. So we have identification of our 4 major bacterial causes of meningitis simply by looking at a gram stain. All right. You can also differentiate these guys as being bacterial versus fungal by a gram stain as well, because you should be able to do a gram stain on fungal cells. Now, you don't know that it's fungi yet, right? You just don't know what your patient has. You do your gram stain on the CSF, you'll look in the microscope and you see eukaryotic cells. So remember, bacteria are smaller and they do not have a nucleus. Whereas eukaryotes like fungi and yeast in this case, right? Whatever, that doesn't want to let me write. Those fungi and yeasts, these guys and helminths and protozoa too, they're all going to be identified as having a nucleus, which is their namesake, right? That's what eukaryote means. And then we have also, they're larger. If they have a nucleus, what else do they have? Organelles, like mitochondria, the Golgi apparatus, the endoplasmic reticulum, all of which can be seen in a gram stain. So gram stain is your key identifier, because also in the gram stain, you won't see anything if it's viral, because you can't see viruses in those microscopes. So if you're looking at a gram stain, you can clearly say whether it is bacterial, whether it is fungal, or whether it is viral if it's meningitis. And if you come to the conclusion that it's bacterial, we can use the gram stain results of whether it is pink or purple, red or purple, however you want to look at that. Red or purple, red is gram negative, purple is gram positive, and then the shape and arrangement of the cells. We always look at this in lab as being this baby stuff. You look at your shape and arrangement of your cells, and like, sure, cool, shape and arrangement. Like, that's not going to tell you anything. You need to be doing PCR. You need to be doing biochemical tests to identify. Not in meningitis. The number one, most important test is going to be that gram stain. And the cell shapes and arrangements, as well as the gram result, is what's going to be helping you determine your treatment right off the bat. You can confirm your diagnosis with PCR. but PCR is only going to confirm. It is not your initial diagnosis. Okay. So gram stains. It's super important that we remember that for this. And believe me when I tell you that when you go to nursing school and you start learning about how to deal with these infectious diseases, this will be something you learn about in the ER setting. Like this is something you will see in an ER setting, because if somebody has meningitis, they're probably in the ER. You don't usually waltz into your primary care physician a week later and be like, I've been having this headache. Is it meningitis? So yeah, so this is stuff you're going to have to be able to recognize off the bat. And that's why they push it so much. It's one of the first things that when I talk about our later stage, like nursing program, because I know a lot of the nursing program professors, and they'll talk about like that's one of the first things they'll bring up. So you do need to know this. You will use it, I promise. So that's the bacterial one. I'm not going to make a separate slide for the fungal. We have these tend to be a little bit more chronic. We have the Cryptococcus neoformans. It's associated with bird droppings and it affects AIDS patients. Then we have Coccidioides immitis. Jeez. Oh my gosh, why? Why? Stop. Calm down. There it goes. Okay. Coccidioides immitis, which is also known as valley fever. It's not only associated with meningitis, but these are fungal and can lead to meningitis. The viral versions are usually enteroviruses that get in from the gut, So ingesting something like contaminated water, like what we see with encephalitis for polio. So gets into the gut. And then a lot of these also are associated with respiratory viruses as well, for children anyways. These are less than 1% fatal and usually resolve on their own. You might get some hospital support like fluids and whatever, IV fluids, but most of the time we're not even going to be touching this with hospital treatments. These are usually a wait it out in your body. If you are healthy, should clear it out on its own. Meningoencephalitis next. This one is amoebas, which are eukaryotes that move by pseudopods or those false feet. That's what defines an amoeba, okay? My dogs are barking. Okay. So you get inflammation of the meninges and the brain, which is encephalo. Okay, so meningoencephalitis, inflammation of the brain and the meninges. We have our amoebas, Naegleria fowleri, which is the cause of PAM or primary amoebic meningoencephalitis. I'm not going to write that down right now because it's too much space. And then we have acanthamoeba, which causes GAM, which is granulomatous. That means there's granuloma formation involved, and that means that we have our white blood cells walling off infections. That's what a granuloma is. They sort of like contain it in like a little round wall of white blood cells. All right, so that's our meningoencephalitis, brain-eating amoebas. That's the detail we're going to go into here and more in the lecture. All right, encephalitis is infection leading to inflammation of the brain itself. So encephalitis is a term that refers to the actual inflammation of the brain. You can have encephalitis and meningitis without infection. Can we agree on that? That can be caused by drugs or injury like trauma. It doesn't always have to be relating to infection, but in our class, we're talking about infection. So encephalitis refers to the inflammation of the brain. And in this case, these are infectious causes of that. So we have acute encephalitis, which is typically going to be caused by arboviruses, which are transmitted by our biting insects. This includes things like West Nile virus. It also technically includes Zika, which we talked about separately from our normal arbovirus infections because it can lead to more serious effects of the fetus, leading to that microcephaly, and it can be quite severe for the unborn child, and can even lead to neurological effects within adults, and even development of Guillain-Barré, which is a permanent effect of having had the infection. So West Nile virus and the things that it's related to. We also have acute encephalitis that can be caused by herpes simplex virus. Meningitis can also be associated with herpes simplex virus. It's usually 2 for meningitis and 1 for encephalitis, but they can interchange there. A lot of times when you have development of encephalitis, you can also develop meningitis, and vice versa when we're talking about these neurological diseases, or not neurological, but nervous system infections. They can lead to neurological effects though. Rabies is caused by a virus. And yes, these are the types of questions I will ask you on the test. What type of pathogen causes rabies? And then your choices might be bacteria, virus, protozoa, fungus. And you would have to know that it's a virus, not just that it causes this encephalitis, or that it makes you foam at the mouth, or leads to hydrophobia, or is 100% fatal. You also need to know that it's a virus. So keep that in mind. We've talked a lot about it, so we'll move on to polio. It's another virus. I would know that the cause of an illness is a virus, a fungus, a protozoa, a bacteria, or a helminth for every single disease we learn about in Unit 4. And I mean it. Okay. So that's a basic thing. So we should be able to say that about these things. Polio, it leads to a lot of neurological problems and muscle degradation as well. It can lead to permanent paralysis, and even after you clear the virus, you can still develop sequelae that will persist and even get worse after you've cleared the virus. So it's bad. We have a vaccine for it that works quite effectively, but it's still present in Afghanistan and Pakistan, which are war-torn, have issues with water purification, and the rural areas, it's just hard to get to these areas to help them with water purification and provide vaccination. Prions, these are not a virus. They are not a bacteria. They're not a fungus. They're not a protozoa. They're not a helminth. These guys are literally only a protein that is misfolded. So can you do PCR to test for these? No, because PCR, polymerase chain reaction, amplifies nucleic acids. Last time I checked, proteins aren't made of nucleic acids. They're made of amino acid chains. Things like phenylalanine and tryptophan and cysteine and alanine and all this stuff lined up next to each other that lead to the folding of this long ribbon of amino acids into these structures that relate to their R groups, that what makes it alanine versus phenylalanine or any of the other stuff. And whether they're charged or not charged, hydrophobic or hydrophilic, leading to our secondary structure of those proteins. That's the initial folding from the guys that are immediately next to you. Then the overall big folding of the overall structure and how the hydrophobics want to hide on the inside to get away from the water of your body and stuff like that's the tertiary. And then those little parts will bind together to other smaller parts to create the functional enzymes or proteins. Those that are the quaternary structure. Yeah, that's coming back up, y'all. Maybe not in the next 2 days, but it will be on your final, which is the next time you're getting tested over this stuff. So you do need to know it. So that is all important to the function of these proteins, as well as how they behave with the environment around them, which determines the cause of the pathogenesis of these prion diseases. They're misfolded proteins, the hydrophobic parts that are usually hidden on the inside of the balled up ribbon. Those guys are now on the outside and don't interact well with water. So they stick together like little droplets of oil would if you dropped them into a big old bowl of water. So they stick together, they aggregate, and they cause holes in the brain that leads to spongiform, looking like a sponge, encephalopathies. So encephalo being brain and pathy being disease. So spongiform diseases of the brain. And these are transmissible. That means they can be passed from individual to another individual. So we wouldn't call it contagious because it's not super highly transmissible, but it is transmissible. if you eat contaminated tissue, so tissue meaning a muscle or nervous tissue like brain, eating that has had these misfolded proteins in it will cause your normal proteins to misfold. And that's how it's transmitted. So this includes things like mad cow disease, Creutzfeldt-Jakob disease, Kuru, which is our cannibal disease, chronic wasting disease in deer, which is probably the most common one in the world, if we're being completely honest with each other. And all of these ones that we don't normally think about in our normal practices, because in our human lives, these are extremely rare, but they're very slow to present. So that's why we call them a subacute encephalitis. It takes decades to see our symptoms as humans. And by the time you have symptoms, it's too late. There's nothing we can do for you if you get this. There's no treatment for this that is effective whatsoever. It's all palliative care. It's 100% fatal. So scary, very rare though. It's all dictated by misfolded protein. There's no PCR test for this. So they're also really hard to get rid of because they're misfolded. They don't behave to our normal everyday cleaning agents the way that we would expect a protein to. So we have to autoclave normally in everyday settings for normal use. We autoclave at about 121 degrees Celsius for 15 to 20 minutes. With prions, you have to go up to 134 or higher in your autoclave, or you have to completely incinerate to ash. It can't be partially incinerated, or you have to use concentrated sodium hydroxide exposure prior to typical autoclaving in order to help get rid of the prion nature of that misfolded protein. It is tough to get rid of these guys. These guys trump every endospore out there. Endospores don't hold a candle to the resistance of a prion. It's pretty crazy to think that it's just a misfolded protein and it can be that serious, but it's true. Not talked about enough in infectious settings. All right, we have tetanus and botulism next. These are both caused by Clostridium species. And these guys, so that means all Clostridium are gram-positive, endospore-forming anaerobes. Always, okay? So they're strict anaerobes. They hate and will die from oxygen. They cannot handle it. So they're only going to express their diseases in deep tissue. This is true for tetanus. It's true for botulism. It's true for gas gangrene. And it's true for C. diff. These guys don't like oxygen. So you might be like, no, because when I get poopy C. diff all over something and somebody touches it, it easily transmits. What are you talking about? They hate oxygen and will always die. Yeah, they're vegetative cells, y'all. Their vegetative cells die. The endospores can survive the nuclear apocalypse. So they don't care. The endospores are what are going to be fine with oxygen. We have endospores for tetanus hanging out in all of your soil right now. Endospores for botulism hanging out in your house probably right now in your regular environment for botulism. The endospores are hanging out, but they're not making you sick right now, are they? Because they can't come to life and make their toxins when they're in endospore form. Because when you're in endospore, you're completely dormant. You do nothing at all. You don't even make toxins. And you can't cause disease without being alive. And dormant means you're not living. You're just waiting to be alive again. So you're not dead, but you're waiting to be alive again, right? So that's the case with all of these Clostridium, and I can't stress it enough. So tetanus, we have a spastic paralysis. That's probably the most important aspect of this one. You cannot relax your muscles. Whereas botulism, we have a flaccid paralysis. I'm trying to find it. Here it is. You can't contract these muscles, okay? So literally botulinum toxin blocks the release of acetylcholine that sends the signal to your muscles to contract. So you literally can't contract them. You die from the paralysis of your diaphragm. You can't breathe. From both of these, tetanus and botulism, your diaphragm is paralyzed and you can't breathe and you die. With tetanus, it's because your diaphragm is seized up and can't relax and you can't get that breathing in and out from that. With botulism, your diaphragm is completely relaxed and can't contract to get that breathing in and out, but the effect is the same. All right, let's go over our commonly missed topics. Chapter 19, our first one is about ringworm. Remember, ringworm is our cutaneous mycoses. Cutaneous is skin. And a mycosis, which is singular, is a fungal infection. Mycoses is plural. So any variation of having a fungal infection in the skin, whether it's in your feet for athlete's foot, or your scalp, or your beard, or your body, or your groin, or your nails. We're talking about the disease of the cutaneous system. So all of this fungal infections of these things are super common. You've heard of them. You've probably dealt with at least some of them in your lives. These are caused by dermatophytes, which are fungi that are involved in the development of cutaneous mycoses. It's really just what it is. And there's 39 different species, roughly, that are associated with these presentations. So these are all ringworm. It's not a worm of any kind. Okay. Okay, so our little memory thing for this one was ring of fungus, not a worm, causes disease on the epiderm. Us. Okay. So it's not a worm. There's a little rhyme for it. First question for this one. Which type of microbe? So this is a very common sort of question that we'll have on the exam. Which type of microbe is associated with the skin disease ringworm? And we've already talked about how it's not a worm. Helminths are our parasitic worms. And this is not what this is, right? Next, we have our protozoa. Protozoa are eukaryotes, just like our fungi are. Cool. So they have their nucleus and their organelles and all of that. But do they cause ringworm? They don't, because we know it's caused by fungus. We just like drilled that into your head repeatedly. So it's not going to be bacteria either. So this is just a matter of remembering ringworm is caused by fungus. I don't know what else to say about it. So next, a patient is diagnosed as having tinea pedis, or athlete's foot, which is a type of ringworm. So right here, even if you couldn't remember that tinea pedis was athlete's foot, I gave that to you. And that athlete's foot was a ringworm, which I also gave you. You should know that ringworm, and we just got it from the previous question, I'm aware, but it's a fungus, right? So we should always be aware of that. So which of the medications below would be appropriate treatment for this infection? I'm giving you explanations of the medications on this one. Do you think you're going to get these explanations on the exam? No. Okay. Because it's a final exam. It is comprehensive. So this is a comprehensive question. I'm not just asking for you to remember that ringworm is fungal, but you're expected to say, okay, if this is a fungus, which is the first thing we know, we've identified that it's a fungus. Number two, what meds are used to treat, TX is for treatment, fungi? Here, I've given you your descriptors. Okay, so we can say and go here and say, okay, well, this is the antifungal one. This is the one we'll choose. But if you don't remember, all of these were on your Unit 3 study sheet and are fair game for me to leave this shit out, right? I don't have to tell you that miconazole is an antifungal. That's being generous. So on the exam, you might get your patient has athlete's foot. What medication are you going to treat it with? And that might be literally the entire question. And your choices might literally just be miconazole, penicillin, ivermectin, metronidazole. So this requires that you know Unit 4, that athlete's foot is a fungus, and Unit 3, that fungi are treated with the azoles, like miconazole, right? It's a good question. All right, chapter 19, concept for gas gangrene. Gas gangrene is caused by Clostridium perfringens, deep tissue infection. It's gram-positive, it's endospore forming, it's anaerobic, found in the soil. It can lead to the cellulitis version that stays local, or the true myonecrosis that can spread within the deep tissues themselves into the healthier tissues. It has toxins that it makes and other virulence factors. Guys, I don't know what to tell you, but I'm probably not going to be getting into a lot of this on our exam because there's just too many things to cover. Also, if I don't talk about it very specifically, like we do with staph, we talked about staph and how you use the coagulase to test for it. And that was a big like whole slide about it and everything. That's something to be aware of. But if I don't actually talk to you about Clostridium perfringens hyaluronidase action, probably not going to ask. Okay. Focus on your big picture stuff. We do talk repeatedly about how Clostridium is an endospore forming genus. This is Clostridium perfringens, therefore it makes endospores. And we learned in our lab, we only tested our gram-positive for endospores specifically because only gram-positives can make endospores. So you automatically know this is going to be an endospore forming gram-positive bacterium. And I have driven it home several times that these are anaerobic bacteria as well. All Clostridium are. Okay. So our memory for this one, gas gangrene can perforate. Just reminding you, perfringens. Okay. Example questions. What type of microbe causes gas gangrene? Same thing as our other question from the last one for ringworm. What type of pathogen? What type of microbe? What type of, overall group would it fall into? Gas gangrene is caused by Clostridium perfringens. If I'm telling you that Clostridium makes endospores, and I've said it 1,000 times in the class, do any of these other guys even make endospores? They don't. I'm sorry. If you think they did, you're not studying well enough. Protozoa, do they make endospores? You might be sitting there thinking, like, I thought they did, though. They do not. They make cysts. These are like endospores, but they are for protozoa. They get their own term. Endospores themselves only apply to bacteria. So if I'm asking you about gas gangrene, this is another way of me asking you about, I could say this, what exact species causes gas gangrene? And your choices could be a variety of things. It would be Clostridium perfringens. So you have to know that, but you better be able to tell me that Clostridium perfringens is a bacterium. You should know that from its name, because we've been talking about these freaking endospores forever. Okay. Why does it require deep wounds to cause the cellulitis or myonecrosis associated with gas gangrene? All right, so gas gangrene, Clostridium perfringens. I'm just going to put C perf so that we remember that it's the Clostridium. We know that this is a gram-positive. We know it's an endospore forming, and we know that it's anaerobic. So why do we need the deep wounds to cause the disease of gas gangrene? Endospores always require deep wounds to be reactivated to undergo germination. Okay, this is something. This is provoking. We'll come back to it. Nutrients. The nutrients they need can only be found in deep tissues. The causative organism only makes its damaging toxins in anaerobic, oxygen-low environments, such as those created within deep wounds, or the organism is sensitive to light exposure. So we've written down the info we need to know about C. perfringens here, right? And we never talked in C. perfringens. This is all we need to know about gas gangrene and C. perfringens. So why would we worry about light exposure? That's not on our list, right? Well, you didn't talk about the nutrients only being in deep tissues either. That's not on our list. So endospores always require deep wounds to be reactivated to undergo germination. Let's talk about, well, we already know C is going to be true, right? Because it's talking about anaerobic. And if you are an anaerobe, number one, Anaerobes are only going to live in the absence of oxygen. They're not equipped to deal with it. So they can't make toxins if they're not alive. So there's that part of it. Also, number 2, deep wounds lack oxygen. Okay. Oxygen also helps protect you from infection from these guys. But if it's deep wound, we can't get the oxygen there and we can't provide that protection with the oxygen. So there's that, right? So we've got this information. But now whenever we look at endospores, always requiring deep wounds, always is our problem here. Anytime you ever see always, you better be sure what answer you're picking on any test in my class or anybody else's. Do endospores always require deep wounds, or do anaerobes always require deep wounds? Uh-oh, anaerobes do. Now, what are our endospore forming genera? Clostridium, which are anaerobes, and bacillus. That should give you your answer here. That's how we know that C is the answer and not A. Because bacillus, even if we aren't talking about how and whether they're anaerobe or aerobe, don't forget they exist. And you should probably know by now that bacillus isn't all anaerobe because they can have action in the oxygen. You can get a pneumo version of anthrax. We mentioned this in class. You can breathe in anthrax spores and develop it in your lungs. There's oxygen there, y'all. They're not anaerobes, and that's not necessarily deep tissue. You can develop anthrax, which is Bacillus anthracis. It just makes endospores. The endospores get into a superficial wound on your skin, exposed to oxygen, and develop that black eschar associated with anthrax. We talked about anthrax. We know about anthrax. We've seen pictures of anthrax. It's exposed to oxygen and it still lives. It is not an anaerobe, and it doesn't require a deep wound. Okay. Our next one is impetigo. It can be caused by S. aureus and or pyogenes. It usually starts with pyogenes, and then staph will come and create the worst aspects of the pathogenesis of the disease. It's usually seen in children. We have exfoliative toxins, A and B, that are exotoxins made by these gram-positive bacteria that lead to development of that flakiness. We can also test for staph with coagulase to see if it coagulates plasma. Usually we use rabbit plasma to test for this. And then the strep bacteria that are associated with this are beta hemolytic, meaning we can get clearing on blood agar. Beta hemolytic, okay. And they also are going to be creating toxins that help with invasion into deeper tissues. So our mnemonic to help remember this: skin ailments impede stripper pyrotechnics. What is this? I wrote this and I'm just like looking at this like, good job, Lauren. You were really creative on this. Let's look at this one because I gotta know what my thinking was behind this. I guess I didn't and finish. I know this is ridiculous. Then I just didn't even finish filling it out. Great. So obviously skin, staphylococcus, as the S of skin is talking about skin infection, and it's also talking about that it's going to be staphylococcus or streptococcus. We've got 2 S diseases here. I is referring to the disease of impetigo, for that's the actual I part of the impede. The ailments is the aureus part of it, to remind you about the aureus. And then stripper is referring to, I'm assuming, strip, because strips of or chains of those cocci. And pyro being pyogenes. So pyogenes, for streptococcus pyogenes, you can think pyro and pyrotechnics to help you remember that it is skin ailments for staph aureus, and then impetigo for impede, and then stripper pyrotechnics for streptopyogenes. It makes sense, I promise. All right, which of these organisms is associated as one of the causes of the disease impetigo? Strep pyogenes. So we already, I'm going back to the stripper pyrotechnics. What was it even? Skin ailments impede. Oh my God, this saying. So you would imagine if you were a stripper and you're putting on this pyrotechnics show that if you have skin ailments, you're not going to get as many tips. I guess that's what I was getting at here. But anyways, here's our little mnemonic. If it doesn't help you remember it now. So impetigo, with our skin ailments impede stripper pyrotechnics, we know our stripper pyrotechnics should be streptococcus pyogenes, and skin ailments referred to staphylococcus. I'm making it work. See, aureus. And impede is referring to impetigo. All right, so here's our choices. What can cause impetigo? It's going to be either this or this. That's what we're looking for. A, yes, Staphylococcus epidermidis is one of your common inhabitants of your skin. Streptococcus saprophyticus is important as far as causing like things like dental caries, but it's not necessarily going to be involved in impetigo from what we're learning about, because it's not Staph aureus and it's not strep pyogenes. So it's A. Thanks, strippers. All right. A culture grown from a 5-year-old's skin sample shows that it has a visible irregular cluster of cocci. So we have our grape-like clusters. So that is our staphylo. We already know that, right? We're coming all the way back, y'all, to unit 1 with that one. So irregular clusters, cocci. So it's a staphylococcus, probably. Round cells, spherical cells, whatever you want to call it. And it's, we're talking about impetigo. So in unit 4, we learned that impetigo is our problematic stripper disease. So we don't want to cause problems for our strippers with this impetigo. And it can be caused per our little mnemonic, Staphylococcus aureus or Streptococcus pyogenes. Right. So this could be Staph aureus for this one, right? Because staph. So that's the information we're given here. Is this consistent? What we're seeing here is the staphylococcus that we're seeing in this sample consistent with impetigo? Absolutely, it is. But why? Is it because, yes, because it matches the description of both typical causes of impetigo, or is it because it matches the description of one of the typical causes of impetigo? Staphylo is not the same as strepto. And y'all, some of y'all, I'm not pointing fingers at all of you, but I'm definitely pointing fingers at some of you, need to remember the difference between staphylo and strepto, because on your little forms you're filling out for your lab exercises, it is very clear to me that some of you don't understand the difference. None of our samples in the lab had strepto grouping, except for some of the very long bacilli, very long hair-like bacilli. Some of them had strepto. And even if you didn't put it on your form, I'm not concerned about that, but you had better not be calling your cocci a strepto, because you will be wrong. And I will dock you points for that. Nobody had strepto cocci. Okay. Staphylo, yes. Irregular or grape-like clusters. That's what the staphylo means. Don't confuse them. Go back and relearn them however you need to. But to me, when I hear strepto, I think of strips like strippers or chains. Okay. And that's what you should be thinking when you hear strepto. This guy has irregular clusters. That's not a strip. It does not cover both. So it's B. All right, next, chapter 20, we're moving into our missed concepts for chapter 20, our nervous system. The first one is immune privilege and what this means. First, we have the blood-brain barrier. It impedes the traffic of substances across from the bloodstream into the nervous system. And we have our own white blood cells that are going to be doing less inflammatory response work in the actual CNS. These are going to be our microglial cells and our brain macrophages. All right, so we've got those. We also have immunosuppressive factors in these tissues that help down-regulate our inflammatory responses. We also have less MHC molecules. If we have less MHC, that means we have less reaction, right, to our antigens. So decreased antigen response, because those are the guys that present the antigens. If we're presenting them less, we're activating less. So that's immune privilege in the central nervous system, and this protects from damage caused by the inflammatory response. All right. So our little mnemonic here is BBB game plan. So BBB obviously is blood-brain barrier. Up here, our game plan. This is going to be referring to all of our immunosuppressive factors that we have going on. So G being our glial cells. A was our antigen presentation, or our MHC stuff that we just talked about. M is going to be our molecules that are involved in the inflammatory response. And then our plan is basically the whole plan of combining all the things to work together to function for that decreased in response. Okay, so our BBB game plan to reduce our reactivity. All right, whatever. Or here's our questions. Which of the choices best describes the function of immune privilege? So without looking at our choices, you should always be looking at what you think the question is asking about. How would we describe the function of our immune privilege? We want to down-regulate inflammation and prevent damage to our nervous system as well, right? So here's our choices. It gets the strongest immune response for the best possible defense. We don't want the strongest immune response. That's what causes the damage. It has no immune response at all. Absolutely not going to help you out if you do get an infection in those tissues. It has a lowered immune response and specialized immune cells to control damaging effects of inflammation. Sounds quite a lot like what we just wrote down. Oh, I don't know why I just scratched that out. Thanks. That's not making it confusing. Come on. And then it has its own lymphatic system. We didn't talk about that. That's not the right choice. Remember that if you have choices on your test that you don't remember that we even ever talked about, number one, if you really don't think we talked about them, it's fair game to ask about. You can always raise your hand and say, Professor Yost, did we ever even talk about lymphatic system being special in the CNS? And I will come over there and be like, no, we did not, because we didn't talk about that. And you're actually coming to me and being like, I'm pretty sure we didn't. I'm going to verify that for you, okay? What role does the blood-brain barrier play in achieving immune privilege? So before we go into our questions, what role does it play? The blood-brain barrier inhibits, or whatever, inhibits is fine, you know what it means. Inhibits stuff from entering the CNS from the blood. This includes white blood cells and other things that are involved in the immune response. So that's my mini summary before I even look at my choices. All right, so our blood-brain barrier, how does it help out with immune privilege? Does it allow immune cells to more freely enter the area? Last time I checked, we're reducing that. So that's not the answer. It prevents the entry of pathogens into the bloodstream from the CNS when the CNS is infected. This is the opposite of the direction that we're controlling. It restricts passage of immune cells and substances from the bloodstream into the CNS. This is sounding more like it. Or does it enhance the immune response by increasing blood flow? That's not part of the function of the blood-brain barrier. All right, next we're moving on to fungal meningitis. This can be caused by, can be caused by two different types of fungi, Cryptococcus neoformans, which is bird poop. You guys see me writing this stuff. I'm just like, that's bird poop and AIDS. That is how your brain should be working. If you hear Cryptococcus neoformans, you need to be thinking, number one, fungus. Number two, bird poop. Number three, AIDS. And maybe number four, these are more chronic meningitis. So you should be thinking meningitis, right? So that's what I think when I hear that one. When I hear Coccidioides immitis, I think number one, fungus. Number two, this occurs in the Southwest, especially Arizona. And number three, meningitis. Great, that's what I know. Our mnemonic for this one, CC in the city. We have Cryptococcus and Coccidioides. It says in the city because we're basically looking at these as diseases that are going to be available in city settings, as well as, I don't say talk about city specifically because I have to write all this stuff down. And it's all helpful. Please read these little notes that I have on here, by the way, because they will help frame these mnemonics in a way that might be more useful than what I can get into here. But some of them apparently didn't fill out, like with the stripper one. That's not helpful for that, but I bet you won't forget it now. Anyway, CC, Cryptococcus, it makes capsules. It's part of the fact that it is resistant to treatment being a fungus. And we also have Coccidioides immitis. So these are all C, 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 C. And then in the city, because we do have actual spread of this one in particular amongst the homeless populations that have HIV and that are exposed to a lot of bird poop in cities, developing these chronic fungal meningitis. So that's fine. We'll move on to the next questions. We have, which of the following is a causative agent of fungal meningitis? Well, we already said CC in the city with all of this. So without even looking at my choices, I should be able to tell you that it's something along the lines of Cryptococcus neoformans. Even if you guys can't spell, you just need to remember when you see them in the choices, but you should have an idea of what these words should be, right? Whenever you're looking at your choices, you shouldn't be confusing Clostridium perfringens and Cyclospora and Coccidioides and Crimean Congo. They're all C's. Don't remember it being C only, right? Because already I wrote down Cryptococcus neoformans. Is that one of our choices? It is not. Our other one that was our fungal meningitis cause was Coccidioides immitis. And that one is. All right, Clostridium perfringens. This is a bacterium, causes gas gangrene. Cyclospora is not associated with our meningitis. This one is a GI diarrhea that is associated with, this is a protozoa. That's what I'm trying to say. This is our answer. And then Crimean Congo, HFV. I put HFV because H is hemorrhagic. So you guys having some real hard time saying hemorrhage. I hope that you guys can get that down for a nursing school. That's a grand majority of you, so don't feel bad if you're one of those people. But when you look at this word, this is just hemorrhagic, hemorrhaging. It's a hemorrhagic disease. It's a disease that leads to hemorrhaging, okay? It's not as weird a word as you guys think it is just by looking at it. Don't let the letters overwhelm you. Hemorrhagic fever virus, okay? All right, which form of fungal meningitis is usually associated with AIDS? So remember when we first started this discussion over here, and I said fungus, bird poop, AIDS, chronic meningitis. So that's our Cryptococcus neoformans up here on number one. So we're looking for that. Here it is. Now remember, Cyclospora, protozoa. We just said that. Clostridium perfringens, bacteria, gas gangrene. And then Coccidioides immitis was a previous question. It is fungal. So we did tick this box, but is it associated with AIDS? No. So Cryptococcus neoformans, bird poop and AIDS. All right, our next one is our brain-eating amoeba, our amoebic meningoencephalitis. Infection of the brain, meninges as well. Okay, so brain and meninges are inflamed here from the infection caused by amoebas. These are Naegleria fowleri, which gets in through like nasal sinus passages. And then acanthamoeba, which is usually going to get in through trauma, often of the eye or any other cuts or wounds. Both of these are found in fresh water. Okay, water. And it's usually warm, fresh water. So by fresh, I don't mean clean. By fresh, I mean it's not salinated, right? So that's our general knowledge group for these concepts, usually through water. Naegleria fowleri is through the sinus cavities typically. Acanthamoeba is through like eye abrasions, like what you have with contact lens wearers, or scratches in the eye or cornea, or open cuts and wounds and things like that. So Naegleria fowleri is associated with PAM, which is primary amoebic. And then acanthamoeba is associated with GAM, which is granulomatous. It's a little bit slower, but not by a lot, and they're both about 97% fatality rate. Here's our mnemonic. Pam's friend gave a gift. So Pam, obviously referring to PAM, primary amoebic meningoencephalitis, associated with Naegleria fowleri. It's very menacing. So, right. So Pam refers to the fact that it prefers active, menacing adventures, right? So Pam's pretty menacing friend. She gave a gift to you, and you probably don't want this gift because it's probably a brain-eating amoeba. And so this is referring to G for gave a gift is referring to GAM, that granulomatous one. The gift being that we have a gradual onset of granulomatous, so it's a slower progression. And it's not something that we want from any of this. Pam, keep your gifts away from us. We don't want your amoebic diseases. So which of the following pathogens is an amoeba? So this is different, right? So I'm not asking you about meningitis here. I'm just asking you which of the following is an amoeba. As we go through the unit, I would hope to all things that are good that at the very least, you could rule out some of these, because we're going to be talking about Neisseria gonorrhea and how it is the cause of gonorrhea, obviously. But that is bacteria. If you didn't get that from Neisseria gonorrhea and the fact that it causes gonorrhea, you should be able to, from this chapter, chapter 20, know that Neisseria can cause Neisseria meningitis. The meningococcus is a cause of meningitis. That is the worst form of meningitis, and that is a bacterium. So we can rule this one out as an amoeba. Amoebas are always going to be eukaryotes that move by pseudopods. We learned this in chapter 5. Yes, we did. So alkylogenes fecalis. Let's come back to this one. Man, Dr. Tim is active today. He sent me like 7 emails already. Okay, Naegleria fowleri. We know this is the answer. So if at the very least you remember the 2 causes of meningoencephalitis, so our PAM and our GAM causes of Naegleria fowleri and acanthamoeba, you know 2 amoebas. Okay, so that's what they are, is amoebas that are causing these things. PAM, the A is amoeba. GAM, the A is amoeba. So if you know their causes, you know their amoebas, and you can find to see, hey, I know these 2 amoebas. Is that in the list? It is. So we've got it. So we already know it's not going to be the other 2. But erythema infectiosum is referring to that disease of 5th disease caused by human parvovirus B19, which last time I checked, a virus is a virus. So it's not this. So that leaves alkaligenes fecalis. And the only right way that you're going to rule that out is either that was your table one's gram negative, right? You either know that because you were at table one, or you would know that alkaligenes fecalis is not Naegleria fowleri or acanthamoeba, are two most talked about amoebas in our entire chapter. Anyways, you should know Naegleria fowleri is an amoeba, is my point. So you should be able to rule this out because you know that this is an amoeba. Which one? Which of the following pathogens is the cause of PAM? So this is our primary amoebic meningoencephalitis. So PAM is Naegleria fowleri, GAM is acanthamoeba, and you're just going to have to remember that. Giardia lamblia, is it an amoeba? No, it is a protozoa though. It has flagella. If it has flagella, it doesn't move with pseudopods. What about trypanosoma cruzii? Do you guys remember what this one is? The trypanosoma? This is the one transmitted by the kissing bug. It is a protozoa. I think we're getting into this one in the next chapter. So it's all in my brain at one time for me. But this is a disease caused by a protozoa transmitted by a kissing bug. This is a protozoa, but this one is not an amoeba, okay? And this is what causes Chagas disease. So this is a cardiovascular disease. It's not going to be meningoencephalitis. It's not neuro. So that leaves us with the choices between these two, which is just a matter of you remembering which one is which. So it's A, Naegleria fowleri causes PAM. Acanthamoeba causes GAM. We're almost done. All right. How many more? Just these two. Okay. These are our arboviruses. Arboviruses are viruses that are transmitted by biting insects. Typically mosquitoes and ticks and fleas and things like that. I guess I can close this and we can see it better. We have West Nile, St. Louis, Eastern equine, Western equine, Venezuelan equine, and then our Bunya viruses like California and La Crosse. And there are even other ones like Japanese and all sorts of Powassan and all sorts of stuff that these guys are pretty tame and super, super uncommon, except for Eastern equine, which can have up to a 70% mortality rate in humans. But these are all transmitted by mosquitoes and lead to neurological effects as well as just general illness. But in most people, most of these viruses aren't going to have symptoms or signs. 70 to 80% of people infected with West Nile won't have any signs or symptoms. So when seeing things, we evaluate very clearly. When being, W for West Nile virus. West Nile virus. Seeing things is referring to St. Louis. Things, this is our Togo viruses, essentially referring to our Western and Eastern equine guys. V is the Venezuelan. And then the C, the clearly, we have C's and L's in this one for California and La Crosse. Don't forget, there's also Powassan and Japanese, but there's a lot of them. Do you guys think the likelihood of me asking you about specifically St. Louis encephalitis, do you think that's ever going to be very high outside of me saying that it is an acute encephalitis caused by an arbovirus? Probably not going to ask you anything else about it, if I even ask you about it. And that's super, 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 super low likelihood. I'm probably going to ask you about West Nile virus because you know it. Okay. And are you going to see any of these randos anywhere else? Probably not, y'all. Most of you are going to be working in Oklahoma. And if you move outside of Oklahoma, you'll become more familiar with the diseases that you'll be dealing with there. But hey, we're in Oklahoma right now. And do you have the likelihood of getting West Nile virus during mosquito season? Yes. Are you likely to have symptoms? No. If you do, are they likely to be severe? No. They're arboviruses because they're transmitted by biting insects like mosquitoes.
So what is a primary transmission method of arboviral encephalitis infections? Arboviruses of any kind, no matter what disease they cause, are biting insects that are transmitting those viruses. So this would include mosquito bites, not any of the other choices. What is encephalitis? Encephalo means brain. Itis, which for some reason is still really hard for people to get when it comes to the exam, is called inflammation of whatever that tissue is. So we're looking for inflammation of the brain specifically, not the meninges and not the nerve. And it doesn't have to deal with always being associated with infection, because you can have encephalitis without infection, right? You can have inflammation of the brain, even if there's not an infection. It could be from trauma or drugs and other things. So we're looking for inflammation of the brain, and that is C. Not infection necessarily. Can infection lead to inflammation? Yes. Can you have infection of tissues and not have inflammation? Yes. All right, rabies. I don't know how people had problems with rabies in my past exams, but they did. You'd think I talk about it enough, but not apparently. So this is a virus. This is a viral infection. You should know that. Okay. It's transmitted in mammals. If you have symptoms or signs, it's almost always 100% fatal. I say almost always 100%. It's like 99.999 or something. There's been like a case or two where people did recover from this, but they did have permanent sequelae associated with it. So you can kick the virus and survive, but it's like extremely rare that that happens, and you aren't going to be your normal self afterwards. Okay. So it's a virus that gets into pretty much any mammalian cell and leads to spread into the nervous tissue. Signs and symptoms are going to be typical illness, then eventually leading to hydrophobia, where you don't want to drink because you can't swallow, not because you're actually afraid of water, leading to foaming at the mouth. We have accumulation of the virus in the salivary glands and therefore transmission in the saliva that cannot be swallowed because you cannot swallow, and then you spread the illness that way. All right, so our mnemonic for this one is raven. So when you think of a raven lying there, essentially looking all dead with other signs of rabies, you can think of the beak of the raven being like associated with the bite transmitting the disease, the black associated with the death effect of it. So you can visualize that raven and see that R is going to be rabies, and that A is going to be acute onset, V, very high mortality, E, encephalitis, and N are neurological symptoms. So that's our visual cue to help us remember that. All right, our example question for rabies: which type of pathogen is the causative agent of rabies encephalitis? Rabies is a virus. And yes, this really is one of my most missed questions on this portion, this chapter for this exam. Please don't forget that rabies is a virus. All right, which of the following is not included as a way to prevent rabies? So this is another thing we need to know about this, right? So we talk about this is from our actual study sheet, by the way. Well, I pull information from that and then I create a more detailed list for you guys. So in our actual study sheet, if we were to go all the way back to it, let me do this, because I literally have the copy of the study sheet up here. So this is chapter 20, right? Rabies, virus, 100% fatal, vaccine available, still prevalent in wild animals. Great. So that's what we needed to know for the study sheet. And the question is, which of the following is not included? Well, we know we need to have the vaccine. So we have vaccination, pre-exposure vaccination, and there's post-exposure vaccination. Okay, so we've got vaccine and we've got vaccine. We know that this is transmitted by the bite of animals. If you don't know that and you need to be learning that from this class, I don't know where you came from, but rabies is a virus disease. It's maintained in the reservoir of animals. So if we can control the animal reservoir by either culling animals that actually have rabies or spreading vaccination within our animals to prevent them from getting rabies, that's another way to control it. And it is related to both the vaccine as well as controlling the animal populations. Do we treat it with antibiotics? Oh my gosh, if you were concerned at all about this question, being like, oh, I don't know if I know, we only said vaccine, I don't know. You should know antibiotics help us all, please. Bacterial infections only. Not viruses. We already said you need to know rabies is a virus. You should not be using antibiotics to treat the actual viral infection. So you can rule that one out all just right away if you were unsure about any of the others. So that's it for chapter 20. We have covered our skin diseases and our neurological diseases. Next week, we will be doing cardio and respiratory. Then we have Thanksgiving. Then we have GI and genitourinary, mostly STIs for that one. In the meantime, I have created a schedule for all of our reviews from here and in between the very end of the semester, of dates, times, and links to Zooms for those. Please attend as many as you can, and we will also be recording them and posting them afterwards as per usual where relevant. Kahoot games where we sit there and you guys play the game to review yourselves, those are not going to be recorded because it doesn't make sense, because it's literally just going to be a screen for people who watch the review. So it's boring. So we're not doing that, but that's how we've always done the things anyway. That shouldn't be news. Does anybody have any questions for me? Let me see.
I have a question really quick, Professor Yost.
Poor Brittany.
Sorry.
You're okay.
On the mecanazole versus the metronidazole. So since they're both zoles, azoles, is there like some, you just have to remember they both start with M and they're both azoles. I'm already like freaking out about how to remember that.
And since we've already moved through unit 3 and then we're bringing this back up again, we want to be sure we have it straight, right? So for our fungal infections, our major fungal control medications are going to end in azole. So think of azole as being your blanket term for fungal infections, but there is only one azole out there that has left the fungal rural, if you will, countryside and moved into the big city, the metro, and that's metronidazole. So metronidazole, think of it as leaving the fungal rural area and moving outside into protozoal infections and anaerobic bacterial infections. So think of, that's how I remember it. And that's stupid, but that's how I remember is they moved to the big metro. Yeah.
I remember you saying that now. Sorry, I forgot about that, but that makes sense.
Absolutely fine. Of course, it sticks with me. I say it every single semester over and over again. So, and we left unit 3 and we kind of are moving with unit 3 behind us for now until we come back and reunite everything at the end. And I get it. So that's your reminder for now. And hopefully that'll stick with you. But that's how I remember it. Yes. Anybody else have any other questions? Awesome. And I know the diseases, I think we're kind of thankful for the fact that a lot of this is memorization stuff for now and not as many concepts. So I hope that helps you guys too in your study efforts. You know, I'm only an email away if you need anything, and we're gonna have a lot of review coming up so that hopefully we can get you guys, have everything straightened out. Remember to put your study sheets together for you with your notes that you need in a format that helps you see it clearly. You can use mine as guidelines for sure for the past ones to help you organize those thoughts. But yeah, really work towards making your own study sheets because they'll work better for you than mine will if you put the effort into them, I promise. So yeah, so I'll leave it at that. If you guys don't have any further questions, and I guess I'll see you guys on Monday or Tuesday. Happy studying.